Hello, everybody. We're really excited to be back here at the museum together,
Yeah.
live into your living rooms, onto your phones. My name is Franziska Settler, and I lead this whole event usually together with my amazing team, who you just had a little look at. We've been at the museum, oh my God, since August 2019. And I think for now, we've mostly done everything online. So I think I'm more trained now in doing the events online, but that means that all of you also watching on YouTube can have a chance to be here with us today, which is really exciting. So we welcome all of you to have a cup of coffee with us or tea. We don't discriminate. You can drink whatever you want. And because it's October, I also brought a pumpkin. I mean, I had to bring one. So here you go. I really like Halloween.
You
so
do, yeah.
I think your topic today also is very fitting
I hope so.
for like spooky month. And yeah, we already had a look at him. It's Miguel. Really happy that you're here today and that you're going to talk about your research with us.
Yeah.
And so welcome. Welcome
you,
again. And I think we've seen each other online quite a few times.
Yeah.
A few times. I now learned that we kind of also attended a live event a few years ago.
Yeah.
I mean, it was just fate that you're here today. So I'm really glad that you could make it happen, especially on a Sunday. And I have so many questions for you. And if you have questions, please feel free to write them either in the Zoom chat. We can also unmute you if you want to ask a question live, or if you're on YouTube, don't worry. Again, you can also post a question there, and we'll definitely answer it. But before it's your turn, I have so many questions. I'm very nosy, so I have lots of questions for you. And my first question is always how you and like how my visitor and how my guest ended up in science. Are you the only scientist in your family?
Yeah. Well, just again, thank you for having me. Definitely, I'm the first scientist in the family. My mom tried to get into science at some point, but as it turned out, she didn't. And then I ended up doing science. I guess I was sort of incentivized by a few teachers at uni. Yeah, so obviously the teaching was only part of their job. They were doing research in parallel. And they kind of once they figured out a few of the students were interested in research, they would kind of motivate us a bit. And there was a couple of teachers which I guess got me into research. Yeah,
that's really nice because
absolutely.
I mean, during my high school years, I was good at biology, so I could do that, and I was good in English and German, but not good in the other science.
Okay.
I never thought I would become a scientist.
Yeah.
I'm a studied paleontologist. I always liked paleontology, and I grew up knowing I wanted to be a paleontologist. But then when people told me, "Oh, you actually have to, be good in like other scientists, not just biology," I was like, oh God, how am I going to do that? So also my family, they're not scientists, so I don't really know how I ended up here, to be honest. But it's good that you had teachers encouraging you.
Yeah, I guess it, I don't know how often it happens, but and even even just one or two teachers can really make a difference.
That's true.
In that regards, yeah.
I think it's good to hear because we had teachers also watch the event in the past. So I think I think it's good for them to hear that they can make a difference. And so where are you from? Are you from here? Because it's in English today. So tell us a little bit where you're from and what you studied during your bachelor's, because that's when you said your teachers kind of encouraged you, right?
Absolutely. Yeah, I'm definitely not German, obviously. So yeah, I come from Portugal, and I kind of moved cities. I have an older sister, and she also moved to Lisbon. So we ended up all living in Lisbon, and she did her studies there, and also I did my veterinary studies there.
You studied veterinary science,
right?
Oh, so did you want to become like a veterinary, like an animal doctor?
Absolutely, yeah, absolutely, okay. Yeah. So that was classic. It's very cliche. You like animals since you were a kid, and then you end up wanting to do veterinary medicine. So yeah, studies in Lisbon, and then so bachelor, and then it's like a 6-year thing. It's integrated master's program. So then
oh, it's combined.
It's combined,
Ah,
yeah.
okay.
So once you finish vet studies back home, you're already qualified to do PhD in case you want to keep doing research.
All right.
So yeah, because it's 6 years. So
okay.
That already qualifies you as a master's degree. But then I also moved to Spain to do a specific master's on wildlife.
On top of that,
Because I kind of wanted to, because in vet studies it's the classics that the teachers, most people want to do clinic, and so they focus a lot on cattle or companion animals, farmed animals, and not so much on wildlife, which was my passion from the early stages of my studies. We also had a small group of students who were passionate on wildlife, and we created like some sort of a wildlife group, and we had also some lectures and invited people working on wildlife. So I went to Spain eventually because I thought they had a really good master's program where I could deepen this interest, and then so I ended up living in Spain for a while. And then I moved back home, and I did clinic, a mixed practice, both companion and small animals, and then also farm animals. That was very exciting. I learned a lot because in vet school, it's always a big group of students, and it's a very practical. It's a really practical study. So then the good thing of doing this, it was about a year that I worked as a vet back home, and then that's where I really learned what it was like to be a vet and be exposed to new situations. And so yeah, and then eventually I quit that job because I got the position here in Berlin, the PhD position.
Right.
So I was kind of moving between Spain, then back home, and now here.
I like I've never heard this, that people call it companion animals. I really like that.
Right. Yeah.
It's much better than pets.
It's I like the term as well. Absolutely.
I'm gonna adapt that now.
It's not that a cow or a sheep or horses cannot be companion animals as well, but we tend to separate companion from farm.
I like
animals.
that a lot. Did you have a favorite companion animal that you worked with?
Well, I'm very biased on this answer because I have a dog at home.
Okay. There we go.
Not here in Berlin, unfortunately, but my parents are taking care of it back home. So yeah, dogs. Yeah.
All right. And you said you came here. So did you ever in the past think you would end up in Germany, or was it just that this position was the perfect one, so that's why you moved?
Right, absolutely. It was mostly about the position and not so much the country or even the city. It just ended up to be Berlin. It could have been elsewhere, I think.
All right. So you saw your like the advertisement for the PhD, and you were like, oh, that sounds like this is for me, and then you applied. And and when did you move here?
That was April 2018.
Oh,
so
18,
that's 3.5 years.
You've been here.
Yeah,
did you learn any German yet?
Well, yeah, ich konnte ein bisschen Deutsch sprechen.
Oh, that's not so bad. Because I know PhD students, they have a lot of work. It's not a lot of time to learn a language.
The thing is, if you do research, everything is in English, and your daily work is going to be in English.
Yeah,
so
I understand.
And that's also true in my case. So you really need to like put effort in parallel, because if you just do your work, you're perfectly fine with English, and you don't really need the German so much if you do research.
And do you want to remind us where exactly you work right now? What is it called? Where is it in Berlin?
Sure, absolutely. So it's the institute is based in Berlin. It's a bit east. We're just next to the Tierpark, Berlin, and so there's lots of collaboration between them and our institute. It's called the Leibniz Institute für Zoo- und Wildtierforschung. So wildlife research in general, and that's why I don't think, to be honest, there's many institutes like this, because literally everyone is focused on wildlife, and this is why it's such a special institute. And when I saw the position, I immediately applied, because it really made sense to try and work there. So it's very fascinating because people work on such diverse species, and there's always interesting projects going on, obviously, and it's very diverse. Yeah.
So this is not going to be a spoiler because I think people are watching, they know that you also work on hyenas.
Yeah.
Is that an animal that you've always been interested in, or because I don't know, I grew up watching The Lion King, and
Obviously,
hyenas were already like a little bit silly
Yeah.
to me. Like, oh, they're kind of like silly animals, and you know when they have this like
Absolutely.
laugh.
Yes, the giggling.
I mean,
yeah.
I like a hyena, but
Absolutely.
yeah, that's how I kind of grew up
Right.
thinking they're funny.
No, I think our well, not only our generation, but even the ones afterwards, it's kind of we have this idea, right, of hyenas being the bad guys, and
Yeah, the bad guys, that's true.
Yeah. So definitely I had no previous knowledge or contact with hyenas before coming here. And it turns out they're extremely fascinating species. Not only from a social point of view, they form these clans. I'll talk about it in a while on the presentation, but so it turned out, so I had no background whatsoever with hyenas.
Except Lion King.
Except Lion King, obviously. Yeah, exactly. And it was not so much, I don't think it was what I'm doing I could actually be doing in any other species. It just happens that the species is so interesting that it's quite easy to collect and gather all the data that we want to answer our questions, our ecological questions. It's a very interesting study species.
Right.
And so yeah, it turns out they're fascinating.
Okay.
Yeah.
And you don't just
Yeah.
study hyenas, you also study other stuff. So maybe you want to show us your slides and tell us exactly what you're doing. So we're going to share the screen, so you can also find out what exactly you're working on. Because I know already a little bit.
Right.
Exactly. Yep.
All right.
And
This one.
This one.
Okay.
And we're gonna do full screen.
Yeah, absolutely.
Again, you can ask questions in the chat. We are gonna have maybe you have to go back to the beginning.
Absolutely.
Oh, don't look, guys, don't look. No. There we are.
Okay, okay.
Now.
I'm sorry.
That's okay. And we can
there we go.
answer questions in the meantime too. So
yeah, sure. Yeah, absolutely. Just yeah, feel free to interrupt. I'm happy to take questions in the meantime as well, or just save them for the end, wherever works best.
Yeah, and Luna, can you see the screen perfectly? Yes. Okay.
Okay.
Well, then we can start.
Good. Yeah, it's working good. Great. Yeah. I kind of split the presentation into two parts. The first half is a little on parasites and how diverse and some life cycles, which in the end I wasn't also parasitology was also always a very fascinating subject in vet school. So I thought it would be interesting to share some facts about parasites here with you guys as well. And then the second half will be a bit more on our work and what we do at the IZW.
I think I already mentioned this for you. I have a love-hate relationship with parasites. I'm very intrigued to learn about this, but I'm always like, oh, parasites. So
no,
I think
absolutely. I think you're not the only one.
This month.
Okay. Yeah, great. So yeah, I kind of gave this title to the presentation because I think it's a question that a few people ask me, and we're going to try to go through it. So the first obvious question is obviously what are they? What are parasites? And the reason I showed this picture here for a start is that what you see here is the very beginning of the life cycle of some parasites, and it's the egg actually of these parasites, or on the top two rows oocytes. And so the first thing I wanted to express here is how diverse they are, because obviously so they all look different. So those are actually different parasite species, and sometimes we can identify them all the way up to genus taxon. just by looking at the eggs. And so that's the first thing I wanted to share, is how diverse they can look morphologically. There's thousands of parasite species, obviously, but already looking at the eggs gives us an idea of what we're looking at. And so they're also very tiny, the egg stage, obviously.
Oh yeah, what is it called? Micrometer, or what is that?
Yeah,
scale?
the scale is in
Tiny,
micrometer.
tiny, tiny.
Absolutely, it's micrometer, exactly. So we observe these in the microscope. We always need a microscope for this. So and the other thing, interestingly, is that if you look on this lower right egg, it's an egg of Toxocara canis. So the correspondent in cats is Toxocara cati. But the reason I'm showing this picture, yeah, it makes sense.
Yeah, cats, that's cute.
Yeah, the nomenclature of parasite species is really interesting in that regards, because very often they're actually associated to the host species.
All right.
So sometimes just by looking at the name, you can already tell, oh, this is a cat parasite. But it also happens that the same species can actually infect different hosts. So that's also quite interesting. And the reason I'm showing this is because it's just an example. There's 100 more of the human impact that these parasites can actually have. So they're called so toxicariasis, the name of the disease, is a zoonosis because it can affect humans, as you can see in the life cycle there. So what happens is if people inadvertently, obviously, eat unwashed, for example, vegetables, and that's why I put the picture right there.
Yeah, thanks.
It happens that exactly. It happens that there's a potential if an infected dog or cat was passing by and defecated, then the eggs were and if this animal was infected, obviously, then the eggs can be shed on the feces, and then if humans inadvertently eat this egg, kids also they're very
Of course,
they
they
like
put
to
everything
play on
in
the
their mouth.
ground.
Yeah.
Absolutely. So it can happen that humans get infected.
So it can. All right. So if I buy a salad and I don't wash it properly,
yeah,
I could get sick.
absolutely. So
oh God.
yeah. so this can definitely happen. So yeah, make sure you wash your veggies properly.
And you probably tell everybody you meet that, like,
yeah,
you need to wash this.
I do it a lot at home. Not just because of COVID. I know at the beginning people were like being super careful on whatever they brought home, and they were literally washing everything. But it's particularly the veggies.
Right.
It's a good policy to.
So still wash it, man.
Yeah, absolutely. So and then the adult stages you can see on the little petri dish also on the picture. So they actually look very much spaghetti-like, and yeah. So they and then obviously they mature in the gastrointestinal tract of the host, and then the adults mate, and then they shed eggs again in the feces. So so it's a closed direct life cycle, we call it, because they only need one species to keep the cycle going. So in this case we call it direct life cycle, and by
and the parasite will stay inside the gut of the animal, let's say the dog.
Yeah.
But it will shed eggs.
It will shed
eggs. The parasite
stays
inside.
Exactly. The parasite stays, and it can really stay for a long time.
Okay.
Yeah.
All right.
And so what I wanted to bring out is that is the zoonotic potential of parasites, and there's many more examples besides this one.
Very spooky.
Yeah.
Very spooky.
So the word parasite comes from the Greek parasitos, which actually means the one who eats at the table of another. And so I really like, it's a very broad definition, obviously, but I really like it because it's literally what they do. They take advantage of the host's resources for their own benefit. So that's why I quite like this definition. And as an example, we from our study group, we actually show this picture quite often. So this is a children that was heavily infected, not with the Toxocara, the one we just saw. They actually look very similar. But this one, this species is called Ascaris lumbricoides, and this species is also kind of a similar cycle, but then it has an intermediate host, and people can get infected, for example, if they eat raw meat from like undercooked meat. It can happen because the cyst, the larvae can be encysted in the meat, in the muscle of the host.
Oh God, in the muscle.
Right. Yeah. Oof. So if undercooked meat has this issue that it can also bring Ascaris problems, for example. And this kid, the reason is that, so actually all these worms were taken from this kid, and he's just showing them all. And the thing is, he was very so his nutrition status was not the best, and he was underdeveloped for his age.
Right.
So
because of that, all the nutrients were taken.
Yeah,
right,
absolutely, exactly. And with such high parasite loads, then it really has a big impact on the host. So obviously, you know, if it's very little parasite numbers, then probably the impact is very little. But it depends on the species, obviously. But you know, with such high parasite burdens, it's really an issue. Yeah.
So I find it interesting because I also, my master's was in biology.
Okay.
And we talked about like symbiosis, but also parasites. And symbiosis is when you have something what you get benefits, right?
Right.
And this is, they have no benefits of being affected by parasites, right?
Right. So obviously, so absolutely. So symbiosis is when both the host and the parasite would gain something with it. There are, so a recent field of studies is actually immunomodulatory effects that parasites have on the host, actually. And there's many interesting studies on a study or human communities that are commonly infected with parasites. And what happens is, if you go to these communities and you suppress and you clear their parasitic load, they start developing other issues, other immune problems.
When the parasite is gone.
Yeah, exactly. So it's a very interesting field of research. It's quite recent, but so this happens that actually having a parasite is not always such, you know, if you have this much burden, it's definitely an issue. But having low burdens, actually there are studies showing that these parasites also modulate the way your gut, well, not only your gut, but also like systemic wise, how your immune system works.
Wow.
And it's not my field of study. I have friends working on that, but so it's typical. So some species, tendentially, you could almost call them as a symbiosis.
All right. Okay.
But
not the ones you look at.
Yeah.
Okay. You only look at the bad guys.
Definitely. Yeah. So definitely, what we're looking in the hyenas is, I won't talk about it a little more ahead, but it's actually there, if there is any fitness consequences for this species. So but technically, the most broad concept is that it's not a symbiotic relationship, it's a parasitic relationship. Yeah.
That kid looks very like chill about all of this.
I guess he's happy because he's just got
been cured
of all of that. Absolutely.
But if I would see that coming out of me, I'd be like, oh God, I would faint.
No, it's it's
child.
a big and obviously it can really lead to I mean, probably if they weren't all cleared out, he would have lots of troubles, especially, you know, the gut can actually be completely blocked by such load. And then if you wouldn't do surgery, then
You
it's
die,
really a
right?
problem. Yeah. Right. So and I just wanted to give a few more examples on other parasites, and this picture probably doesn't tell you much at first, but so what we're looking at are erythrocytes, red blood cells, all these little purple circles there, and the ones that are that have little darker purple on them, they are trophozoites from a parasite called Plasmodium, and Plasmodium is the cause of malaria, which you've certainly all heard
That's
about.
how I've heard of this before.
Yes.
I recognize the name.
Right, exactly. So it's a little a complex life cycle for malaria. It's a bit simplified in here, but so it has a liver stage where it multiplies, and then there's a red blood cell stage, so systemic stage, which was what we're looking at in the picture. So and the reason I brought this Plasmodium parasite is not only that's the reason why mosquitoes actually outrank humans in being the world's deadliest animal. So the size of the red circles is proportional to it represents the number of people killed by animals per year, and obviously the bigger the circle, the more deaths. And so the reason I really like this graph is not so much because of mosquitoes, but and malaria, but it's actually that all these other 5 circles that I'm showing here, they are there because they are a parasitic. They're causing a parasitic disease. And so in the case of tsetse and assassin's bug, they transmit trypanosoma, which is a blood parasite as well, such as Plasmodium. And freshwater snails, they are the intermediate host for a parasite called Schistosoma, which is living in the bloodstream. Also, it's also a worm, but it's living in the bloodstream of humans, which is very gross, very tough.
Yeah.
So the names are a little small, but I guess you can read them. Schistosomiasis is called, and it's a disease. So it's a freshwater snail, and what happens is these people get infected by going into waters that have these parasite, and actually the parasite can penetrate through the skin.
And they live in, so the host before it comes into a human is a snail?
Yes, it's a snail.
They come out
of the snail. They grow in the water, and then they infect humans as they go in the water.
Interesting, because snails are so different.
Yes, absolutely.
The smallest.
Which is why I love parasitology, because there's literally a whole world to explore as far as different hosts. And the last two at the bottom, Ascaris and tapeworms, they're also quite common in human and actually killing lots of people. So that's why I love this graph so much, is that parasites are actually having a big impact on many communities. And so if there's something you take from this talk, is that, you know, snails kill more people than sharks and wolves and lions and elephants, because there's actually lots of people being affected by not only the snails, but, you know, all the other parasitic diseases.
To hear like a snail. I mean, I'm most scared of snakes.
Right. Okay.
So snakes are up there. I see them.
Right. Exactly.
And I know at least one person who watches who definitely also scared of snakes.
Okay.
And this confirms my fear. I'm just saying.
Yeah.
You know,
putting the snakes right up there.
And it puts snails also on the map now.
Yes. Okay. Okay.
Being scared of snails.
Yeah.
But I assume the snails.
Don't kill one if you see it, though.
Of course not.
You would never
avoid
do that.
them.
Yes, just avoid them.
but also these animals, the tsetse fly and assassin's bug, that's a pretty cool name.
It is a pretty cool name, and it's really because, so the names, so sleeping sickness and Chagas disease, they're actually causing very similar diseases, this trypanosomiasis, but it's just that they're geographically differently located. So the tsetse flies, they're in Africa, and they cause the sleeping sickness, and the assassin's bug occur in America, mostly Latin America, South America. But the funny thing about these two diseases is that the reason it's called sleeping sickness on the tsetse flies is because one of the symptoms that people get really tired and very, very sleepy when they're infected with trypanosoma. So it's actually the species is actually also different, but it's also the concept of the disease is the same. It's just different geographical regions. Yeah.
Sorry to interrupt. There is someone who would like to ask a question, and so it's okay for you guys, or maybe at the end. It's your decision.
Yeah, good. Sure. Yeah.
Sure. Okay. So I will unmute. So you can ask.
Hi. So I was just wondering, since I think I figured it out by now, since you talked some more, since I had the question, but I'll ask it anyway.
Okay.
I was just wondering, how is the parasite not very harmful to the mosquito, or how come does the mosquito not die and can just go on infecting other people? Like, why does it not die when it's itself infected by the parasite? I think, now that we've talked more, the answer might be that parasites are not designed to kill their host, but they're designed to just eat. Like, they harm their host, but they can't be deadly, because if they're deadly, then the host is going to die, and then they're not going to get any more nutrients. So they're probably somehow, like, evolution designed them so that they are not 100% deadly, so that their host can still keep living and eating. That's probably, the reason why, but I just would like to hear your thoughts.
It's a good question. Is that true?
It's a beautiful question, and so it's absolutely true what you just said. So this concept that the parasite, or in general, actually we can extend this concept to all other pathogens. In fact, that it's not so beneficial to kill your host, right? So technically, yes, this is very much true. It happens for parasites, but also for viruses and bacteria. If you're a virus and we're in the middle of not in the middle, I hope anymore.
Yeah, let's hope we're towards the end of the pandemic.
Yeah, so if you're very deadly, then you either have to, either you spread really quickly, or you're going to be in trouble, right? So because you're killing your host. But in the case of parasites, and you asked about malaria, about the mosquito, it's also true for the other vectors. It's that, for example, for malaria case, what happens is the mosquito, the way the mosquito gets infected is if it takes a blood meal from an infected person with malaria, obviously. So it takes in the parasite, and then this parasite actually replicates, but then it's located in the salivary glands of this mosquito. So it's very quickly transmitted when the next blood meal happens. So it's definitely not killing the host. And there are studies on how the mosquitoes react when they're infected, because vaccine, and I think I have a slide on it just in a few minutes time. Obviously, because for vaccine development for malaria, it's a huge thing, and apparently we're a little closer to getting a decent vaccine for malaria, but so there's people literally working their whole lives just on the mosquito, because it's so complex, and but definitely it's not detrimental for the mosquito, and also the other cases that we're looking here. Also, for example, the Ascaris roundworm, the worm that affects humans is called it's either lumbricoides or Ascaris suum can also happen, and suum comes from pig, and so they infect pigs, and hence also the raw meat.
Right.
But for example, in Ascaris, what happens is the larvae insist in the, I just mentioned it a while ago, the larvae insist in the muscle. So the previous stages of the parasite, they insist. So they form these cysts in the muscle, and there's absolutely no no problem for the animal in this case. So yeah, yeah, it's definitely not a big problem for the vectors. Yeah.
Right. Good question.
Yeah, it's a good
Thanks
question.
for
It's a great
asking.
question. Yeah.
All right.
Do we have any more questions? Probably not. Okay.
I don't
If we
think
do,
I don't think we have any
yeah,
in
Luna
the,
can just
chat,
tell us
so
in
you can
case.
keep going.
Right. So yeah, this was just a little also curiosity that it's this paper. It's open access, so anyone can read it, and it was published in Lancet earlier this year, and it's just about a new vaccine trial that for malaria that is apparently more promising. It was just a curiosity. So yeah, moving on to this other very interesting parasite, and I'm sorry for the very visual pictures of poop. We do work with poop a lot if we want to study parasites and if we are parasitologists, because it already tells us a lot about the individual and the status of their gut and whatsoever. So the reason I wanted to show this is that so you can see on the oh, I think they're gonna see my arrow. On the left picture, there's a little white thingy on the feces that if you have a dog, keep your eye open for this, because this this species, and I'm gonna play a little video here. Oh, I'm sorry.
See if it works.
I hope it does. Okay, here we go.
Oh, there we
so
go.
this it actually moves, and
Oh, I don't like
it's
that.
very fascinating.
I don't like that.
I did already for a few times find this in my dog's poop,
What?
actually, and I'm a parasitologist. So even and we deworm our dog, as everyone should do, but still, you know, parasites are evasive, and the deworming also sometimes is not 100% effective. So and the interesting stuff about this species is that what we're looking at, this white, we call it in the vet studies, we say it actually resembles a grain of rice. That's what our teachers tell us to help us memorize. And it moves, and so what we're looking at is actually just a little segment of the parasite that was released. It's usually the more distal part of the parasite, and they release the segment because,
so at the end.
Yes, at the very end. And so on the and the reason they release this is because it's full of eggs. So you can see on the dark with the dark background, the picture, the little white circles that
dots.
you see there, the dots, they are actually egg packs, and each of them has around 7 to 10 little dipylidium eggs. Dipylidium is the name of this parasite. Dipylidium caninum in this case. It's a dog parasite, and the reason I wanted to bring this up is not only because it's very pretty, but because yes.
Okay, well, that's debatable.
Yes, it's debatable. Yeah. But the reason I wanted to bring this up is because this is an example of a little more complex life cycle where similarly to malaria, it also has an intermediate host, which is also an insect, in this case not a mosquito, but a flea, which you can see on the left side, the red arrows of the life cycle. So the fleas take up this egg pack, which you can see on the lower part of the life cycle. They eat it up, and then the dogs or the cats, because it also infects cats, the dogs or the cats, because they're very itchy, they actually, if you've ever, do you have dogs, Francisca?
No,
okay.
I don't. But I know people with dogs.
Okay, right, exactly. Make them watch this
I
presentation.
will.
So what happens is that they're very itchy if they have fleas, obviously, and we can see often when animals are infected with fleas, they bite a lot because they're itchy.
Mm-hmm.
But also what happens is they eventually ingest some of these fleas.
Mm-hmm.
And what happens if they ingest the flea is that they won't actually, the parasite will mature, will end its maturation inside the dog or the cat. And that's how my dog got infected, because it happened already a few times that he had some fleas. So then he ended up having this parasite.
He never learned.
Yeah, absolutely. He didn't learn the first time. Yeah.
Also, we have something in the chat. So if you click on the chat, I think we have.
Oh
some
yeah,
questions.
absolutely.
It popped up right there.
Let's have a look.
Let's see.
Oh, okay.
Oh yeah. So someone asked on YouTube, are there studies on which countries have the most deaths caused by parasites?
Oh, okay.
Because you said some of them, or like the tsetse fly is in Africa.
Yes.
So, and this was quite a big killer, right?
And, yeah.
So are there studies on where we have the most?
Sure, absolutely. In terms of deaths per human, so this is definitely very, very much monitored, because it's if it's a human dying, it's a big thing. Not so much on, obviously, yeah. So on as far as the the veterinarian part of the job is concerned, it's not so common, to be honest, to see animals dying of parasites. There is a very deadly parasite in dogs, but now there's already a vaccine and also treatment, which is called Leishmania, and actually it also infects people, but it's very depressing. But so Leishmania is probably the most deadliest parasite in companion animals, causing the deadly parasite in companion animals. But then all the others, they're quite controlled, because these animals tend to be regularly dewormed. But as far as humans are concerned, definitely there are studies, and many of them are actually open access, so anyone can look it up. For example, on Google Scholar or on PubMed, and there's many very descriptive studies on the deaths caused by parasites in people. Obviously, it's true that most of these diseases that I've mentioned so far, as far as humans are concerned, they happen often more in the tropic areas, or
like the global south more.
Kind of in between the tropics, and also unfortunately where hygiene conditions are not ideal, are not the best, as we are just privileged that we live in Europe in our case. But if sanitary conditions are not the best, then obviously it's a big problem. And this graph, the first graph with the red dots from malaria, and on the others, obviously most of these deaths tend to occur in underdeveloped countries that don't have the best conditions on best sanitary conditions. Although obviously we can also find all these diseases in people with better that live in countries with better healthcare and sanitary conditions. So obviously there are cases of these zoonoses elsewhere, and they're more easily detected, obviously, and they're usually not causing so much deaths per se, but definitely many studies are describing this. And
all right.
Yeah.
Okay, thank you.
Yeah, was that the only one we had in chat? I think so.
I think the other one was asked in person.
Okay. Yeah.
Okay.
Great. Cool. Yeah, and so just another fun fact of this parasite is the scientist there on the upper right is holding the the whole adult parasite, the whole adult Dipylidium. And the fun fact is that this was used already in Victorian era at times, that these parasites were used actually for the case of tapeworms in general. So this Dipylidium is a tapeworm. It's one classification of parasites. There's many other tapeworms, but Dipylidium was used actually Diphyllobotium, it's a similar parasite, but it was used for weight loss, actually. So
and it says safe for kids on the
yeah,
package. It's horrifying.
It's horrifying. It's a bit of poetry right there, what they write actually on the package, and you know, they write, get rid of life-threatening fat and without exercise.
Eat as much as you want.
Absolutely, yeah. So these friendly little creatures can reside harmlessly inside your body and consume your extra calories. So it's actually very fascinating. So to some extent, it's true, because, you know, as we mentioned before, if you have very little loads, then they're kind of harmless, but also it's probably not the best idea to on purpose eat the tapeworm.
I'd be worried,
so you can get rid of them,
that this small load becomes a big load,
right?
You know, because how do you control that?
Yeah, exactly.
Okay.
So just to say that it was used yeah, in the past. I think it's not so common anymore, but still, it's a fun fact about this parasite in particular.
Right.
Yeah. So, oh, I guess I need to click again, right? Because
yeah, click on the slides and then
chat. Exactly. Okay, there we go. So this one is also concerning, again, since we just talked about fleas and how interesting they are. This is another example of not a parasitic species, or so what we're looking at on the picture is, so the cells, it's once again a blood smear, and the cells, most of them, they're neutrophils, but the little round and light red things you see there, they're bacteria, and they're called Yersinia pestis, and the bacteria is the cause of plague. So for sure, all of you have heard about plague, but the reason I brought this up is once again because even though, you know, Yersinia is a bacteria, the flea has to be involved, and the flea is a parasite. So here we come into and that's why I keep saying that it's so diverse, because not only the parasites can be you know, the pathogen itself, but also the parasites can be vectors of other stuff like bacteria or even viruses. So yeah, so there's been a few outbreaks of plague in history. The fall of the Byzantine Empire already in the 6th century was caused by a massive plague outbreak. There was a plague outbreak in the 14th, 15th, I think 14th century in the Mediterranean basin that also wiped out many people. And the most recent one happened in downtown LA, 1924, I believe. And the reason I show a little bit of this history of this last outbreak is because it was still, so people kind of suspected at the time, oh, this could be plague, but people were not so familiar with it, and also it's Los Angeles, and the authorities were not so keen into saying, oh, we have a plague problem.
Right. Yeah.
So
I mean, it's scary. That word.
It's scary, exactly, because you know, you don't want to be in charge if a plague outbreak happens in your city.
Yeah.
So, you know, people try to kind of the story is very interesting, and there's a few books about it, but it was kind of funny because it happened in this place in downtown LA where it was a place where many where many Mexicans lived. It was kind of a Mexican quarter or neighborhood. And then what happened, interestingly, is that the authorities, they didn't want to make it public that they have a big plague problem. Also, they were not sure at the beginning, but also what they did immediately was they put this neighborhood into a strict lockdown. And what happened was, you know, maybe they did it because, you know, they're Mexicans, and, you know,
Prejudice.
yeah, a bit of prejudice. But the funny thing, it turns out, is that it actually worked very nicely to contain the outbreak. And so, yeah, the sanitary conditions, again, were not the best. And what happened was, after much investigation, they realized that there was a big problem. Many of these houses had a problem with rats.
Right.
And what eventually they did was they screened the rats for fleas, and they found that the fleas were infected with the bacteria, with Yersinia. And this was the first time where it was very neatly described where the bacteria was coming from and what's the problem. And in this case, the problem were the fleas, and they still are the problem, because what happens usually is that you can see on the life cycle on the upper right of the slide, that usually the fleas, they have, so the Yersinia plague, it has a sylvatic cycle. So in the wild, often wild rodents like prairie dogs and stuff, they're infected with Yersinia, with the bacteria, but they have no consequence. It has no harm to them. And so the kind of the plague is kept, and it's very hard to get rid of it, virtually impossible, because it's kept in these prairie right. dogs, where, you know, very far from the cities, and so that's why it's very hard to get rid of it. Sporadically, and that's what happened in 1924. What happens is, if there's big die-offs of wild rodents, or if for some reason the wild rodents are coming closer in contact with people, either in urban or more outskirt environment, then what happens is the fleas eventually also get in contact with the rats and the domestic
right.
mice and rats that are then really close to humans. And then eventually the humans can get a problem if they have a big rodent problem in this case in the city.
And what's really funny is, I mean, it's not this specifically, it's not funny, but when I used to live in America, and whenever I saw like a prairie dog, I think they're so cute.
They are.
And I love them.
Yes.
But Americans hate them.
Okay.
They hate them.
Maybe that's the reason.
Maybe that's the reason. And
right.
whenever I'm like, oh my God, look cute, they're like, let's shoot them. You know, like some people actually like shoot them.
Right.
Not in, you know, downtown LA, but in, you know, they just don't like prairie dogs at all.
Absolutely.
They also ruin gardens, fences and stuff. But whenever I saw one, I'm like, oh look, there's so cute. And they're like, no, they carry diseases. I'm like, oh, okay.
Absolutely. That's I'm pretty sure, because, you know, in the communities they're very aware that, I mean, it's 1924, it's less than 100 years ago. So it's actually not a very far distant past. And people know that it's, you know, to be honest, here in the cities, we also, you know, whenever we looked at rats and mice, many people think the same, right? And they're kind of...
the camera turned off again. You don't see us anymore, I think.
Oh, okay.
I don't know what we did. Let's just keep going, because the slides are visible.
Okay.
Maybe our team is going to help.
Okay.
But for some reason, it turned off.
Okay.
But you can still hear our lovely voices.
Still hear us. Okay. Wonderful.
Let's keep going.
Okay.
Yeah.
Yeah, and also so in the plot there, it's also very interesting, because the full line is the number of infected people, and the dotted line is the number of deaths. So actually, you can see very neatly that the deaths occur very few days after the infected, and they're almost the same. So it's a very deadly disease. So because the peaks are actually the same height,
just delayed.
Just a few days delayed. So that's why I brought this here. And obviously there's many books on the topic. Epidemics in Society talks about Black Plague. Of course, it's actually the cover page picture. So Yeah, oh, actually, this is a good example of a study that actually shows the history of plague and the number of cases reported. So this is actually an example for the question we had before, studies country-wise, what's causing the problems, and here we have an example of that. Yeah, so moving on. Oh, I think we have the.
I think we're having a little helper helping with the camera, but I think we can keep looking at the pictures anyway.
Okay,
if you hear some commotion, it's probably the lovely Luna who's helping us.
Yeah. Oh, I see she comes.
There she is. But yeah, what is that? Is an ant?
Yeah. So this is the last example, and then we'll move on to the second part of the presentation.
The hyenas.
Yes, absolutely.
We promised hyenas.
Yes, but I still wanted to. I still wanted to bring this up because it's my personal favorite parasite.
I was going to ask what your favorite is, but
yes,
I guess you answered already before I could
there's
ask.
your answer. So what happens is it's called the Crocellum, and it's very fascinating because again it has a snail host like the one we mentioned before, but in this case it's more fascinating even because so the usual main hosts are cattle, but what happens is the snails can get infected with the embryonated eggs from this parasite, and then the parasite keeps developing inside the snail, and then the Ascaris, the next stage of the parasite, is released on the slime of the snail.
Okay.
And then what happens is that the ants actually feed on this slime left behind by the snails, and if the slime has the parasitic stage, the Ascaris, then they developed into Metascaris. And here the beauty happens is that since the parasite obviously wants the cycle to move on, the parasite actually takes over the ant behavior.
Oh, I've heard of these.
Yes, and the ants get affected by this, and they change their behavior, and they move up in the grass, so that it's easier for the cattle to actually eat it. So it's really a behavior a modulating parasite here, and that's why it's my personal favorite.
It's the creepiest one.
It's the creepiest. And so there's obviously other examples of parasites literally taking over the host behavior, and this is just one of them. Yeah. So yeah, and I think this kind of concludes the first half of the. I just wanted to show this very last one before we move on. That this is how we kind of classify the parasites into ectoparasites and endoparasites, and so we have fleas, ticks, and mites, and also acari as ectoparasites, and then a whole bunch of endoparasites. And I think this kind of concludes the first half.
All right. I think hopefully people can see us again.
And that was.
I'm just yes.
Yeah.
There we go. We're back.
Okay.
I had to turn the video back.
Yeah.
That's really cool. Also, ticks are some of my biggest. So I used to do a lot of field work. Thanks again.
Okay. Yeah. Thanks, Luna.
Lots of field work, and whenever I saw a tick, like on a shoe or on a person, I would bolt.
Yeah.
I couldn't concentrate anymore on fossils or on rocks because I'd be like, there's a tick, I need to get out of here.
Of course.
I'm so scared of ticks.
It's a big thing. There are big issues. There was, for example, the one I had close contact with was when I was doing my master's in Spain. There was this one death caused by a person was bitten by a tick, and it turns out this tick was infected with Crimean Congo fever.
What?
And so the tick, so actually this person was infected by the tick and it died. Sorry, not the tick died. The person
person
died
died.
from it because it's a very harmful disease. And what happened was this actually triggered, when I was there at the time, it triggered a national surveillance program for Crimean Congo fever because, and so then from then onwards there were massive screenings of ticks in the field to check for this fever. And it's a big thing. Ticks can be a big problem, also because they don't really suffer from the parasite. Again, they just survive and they keep transmitting it all on and on. So, and besides that, there's borreliosis, Lyme disease. There's all different kinds of Rickettsia like bacteria, and so it's a big, ticks are a big issue. And obviously for people who go in the field more often, but still, yeah.
All right.
Yeah. So.
Oh, and you said so now we learned about parasites and what they are and what kinds they are.
Yeah.
And but you work on specific animals in combination with ticks, or do you work on, because you still work on hyenas?
Yes,
right? So,
absolutely. Yeah.
Parasites,
Absolutely. My project is not so directly related to ticks, but so there are. Well, for example, we do have closely, or we have collaborations with the veterinary school from the Freie Universität, and they, for example, they do lots of they screen, for example, for Babesia, which can also infect domestic animals, and they screen for many tick-borne diseases in wildlife. So not me directly, but there are many people working on that.
All right.
Yeah. So yeah, it's a whole world. and this slide kind of sums it up a bit. So I guess we can then probably move on. Oh, I'll click again on the slide.
Now that people can see us again.
Absolutely. Good. It's good to be back. Oh, and yeah, I had this kind of a spoiler. Well, not a spoiler, but so the Naturkunde very interestingly had an exhibition on the life of parasites and life undercover. So it's apparently being restructured somehow, the exhibition, but so for anyone living in Berlin, possibly you visited, and I think they also had this kind of a virtual that you could visit the exhibition virtually. I suspect during pandemic.
I'm not 100% sure, but I know of this exhibition.
Yeah, right. Yeah. Exactly.
It's a very striking image.
It is.
So like it is. If you see it somewhere, you're like, oh god.
Exactly. So I believe it's an electron microscopic picture, perhaps of an Anopheles mosquito. It looks so.
Oh yeah, it looks like a mosquito, right?
Yeah, it's definitely a mosquito. It has a big proboscis. Yeah, it's and it's an electron microscopic picture. So yeah. So what do we do?
Yay, that's hyenas.
Yes. So my project is funded by the Deutsches Forschungsgemeinschaft, the DFG, and it's so I'm based at the IZW Berlin, the Leibniz Institute for Zoo and Wildlife Research. But my project is so I'm part of the research training group called Parasite Infections. It's a research training group from the Freie Universität, and we have different colleagues working on different species of parasites on different hosts. And yeah, this is so it's a FU research group, and what so a little bit on hyenas. They are, as I said, so the word in German is Tupfelhyäne, and they're as I mentioned before, from my perspective, who had no idea about hyenas before, they turned out to be quite interesting because so they're very social species. So they form these clans, and what happens is they have each of the clan territory has a den where they keep the cubs, and where most of the social activity of the clan happens in the very vicinity of this den. So they live in groups, and they're kind of in the same area. So they change dens sometimes, and they change regions, but it's very it's quite easy to keep track of them once you kind of know where the clan is, because they don't move around so much. So because they're always the den will always be there, and the cubs will always be there, and even if they have to often. So this is I think I have a slide on the Serengeti National Park, which is our study system, but the what happens is because they prey large herbivores. They sometimes, and the Serengeti is very famous by the large herbivores migration. So sometimes actually they have to the females have to commute long distances to actually find food, but they always come back to the den where the center of the whole clan is happening. So it's kind of easy to keep track of them once you kind of know where what's the clan's territory.
So you don't need to have tracking
Absolutely
devices or
not.
anything?
Yeah,
No?
absolutely
Okay.
not.
Okay.
We exactly. So there's no collars, GPS collars in our study clans in particular. We don't need them because I'm obviously not in the field, but our field veterinarian, she's monitoring them, and she once she knows where the clan is, where the center of everything is happening, then she always goes back there, and she knows where they tend to be. So that was kind of a bit of the left picture on the slide, and the middle picture is that they form. So they have a very, not only are they a very social species, but also they are a very ranked. So they work on the basis of social ranking. So social status is very important in this species. So what happens is it's a female dominated society. Absolutely. So
the women are in charge.
Yes, the women are absolutely in charge. They so every female outranks every male, and obviously there's also a gradient in the females, and there's a ranking in the males, but all the females are outrank all the males, and the ranking happens on a basis of, so you can see on the picture, it's actually, so you see two dominant individuals. They have their tails up and their heads, sorry, their ears leaning forward. So they're two dominant individuals, and you have a subordinate individual with the head down and and the ears towards the back, a bit like what we see in dogs. This subordinate position, and so they work on a basis of coalition, and there's lots of social interactions going on, but yeah, so it's a very stratified system, and what so this is actually the key for everything, also for what we study, because their ranking actually determines their priority of access to food. So what happens when they hunt, and perhaps we can already go on talk a bit about the picture on the right side, is that actually they hunt over 90% of what they eat. So they are, yeah, so people tend to have these misconceptions that they are scavenging.
Yeah,
right?
That's what how you picture them, they eat the leftovers.
Yes, exactly. So also I guess from the lion,
of course, the lions
because
they
live in this elephant graveyard and stuff in the movie. So actually they're not scavenging as much, and they do scavenge, of course. I mean, who doesn't want a bit of easy free food, but they actually hunt a lot. And most of what they eat, they have to hunt for it, and they they hunt in groups, but they can also hunt separately, and they so they mostly hunt. So in the picture it's a buffalo, but so their jaws, they're actually, it's so pretty to watch them in the wild. Or also, if you want to see them and you've never been to the Deer Park, there's also hyenas at the Deer Park, so you can also see them there. But it's very impressive to actually look at the anatomy of their skull. Actually, this is very interesting for you as well, because their jaws are very, very powerful. So their skull is actually tremendously powerful. And they either hunt, so they mostly hunt zebras, Thompson gazelles, but also wildebeest or gnus in English, but also buffaloes, and they're perfectly capable of bringing down, 3 individuals are perfectly capable of bringing down an adult buffalo. Yeah, so and the priority of access to food. So when they hunt, and we go back to the ranking. So when they hunt, the first to feed are the highest rankings and ranking individuals. So then once they're satisfied, well, sometimes you see these videos of them like kind of feeding in a carcass, and there's many hyenas feeding at the same time. So that's kind of okay, you know, there's plenty of food, but still then in these cases also the highest rankings, they have priority of access to food, and once they're satisfied, then you know then the lower ranking individuals they can take the rest and they can feed. But the biggest or the most the highest quality, let's say, put it like this, meat goes to the highest ranking individuals. And then here we have a picture that's the map of the Serengeti, and yeah, so what I was mentioning before, that the arrows there, they represent the migratory patterns of the big herds, and on the lower picture it's a herd of gnus. So that's their main prey actually. And of course, you know, there's a whole there's cheetah, giraffe. That's just a few pictures that I took during my time there. So it's a very diverse ecosystem, obviously, and that's where our study clans are in the very north, actually, are based. Yeah, they're based at the north of the Serengeti, in the border, so in Tanzania, but so close to Arusha, but in Tanzania, not far from the border with Kenya, and from Lake Victoria. Yeah.
Sorry to interrupt again, but we have again a question, and somebody want to unmute.
Okay.
Oh yeah, sure.
Here with you, or
Great. Yeah.
Yeah, go ahead.
Hello. So this is a question I had a bit of a while back. So it's still on the topic of parasites.
Okay.
I was just wondering that since parasites come in different sizes, is it possible, first of all, is it possible for a parasite to infect another parasite? And second of all, since it seems to be pretty efficient to be a parasite, how come we did not all evolve to become some sort of parasite?
I mean, I've seen the movie Parasite. Some people
Yes.
have these tendencies. But it's a good question. Like, are there parasites?
It's a good
other
question.
parasites?
So this question is it depends on what you define as a parasite, because for example there's many for example there's fungi, and there's which you know, they are parasitic pathogens, but in this case, even though it's not parasite as we consider it a parasite in the veterinary studies, it is still a parasite, because it's still taking advantage of the host only for its benefit, because there's, you know, I can't actually recall the name right now, but these, for example, these fungi that infect insects, and these insects are also vectors for other disease, or so they're also transmitting other parasites or other disease or the other pathogens. So it is possible that you have, you know, parasites infecting other parasites. Do I recall any particular example of this? I don't. I'll think about it. It's a very good question. There are parasitic cases of parasite species. Yes, definitely. I'll try to think of a clear example for this.
We still have some time.
Yeah, I'll think about it. But the answer is definitely yes. How come did we not all evolve into being a parasite? Well, that's probably even a harder.
That's a hard question.
That's a hard question because
we're capable to live on our own.
Parasites are definitely very primitive organisms, and they've been around for forever, literally. But
it works for them.
yeah, it does. It's tricky. I don't know why. I guess then parasites still need their hosts, right? So if there were no hosts, technically, we humans, as we saw in the cycles, we can be we can be infected by these parasites, but very often what happens is the main host for these parasites, it's not actually us.
We just like a stage.
Yes, exactly. So the main host for these parasites was there way before us humans were. So actually, the parasites, they didn't even need the humans in the first place, right? If we put it that way. And obviously then with time, since the primitive of humans, then species who were not using humans obviously as the definite host, then they once they jumped into humans, then they evolved and they split into actually now species that only infect humans. But technically,
parasites, then
technically they don't need us, right?
Don't need us, yeah.
So they were there way before us. Right. I'm sorry I cannot answer that more neatly, but I guess that's what it is.
But it's a very clever question, so
it is.
It's difficult to answer.
It is, yeah.
Can you check if we have any more in the chat? Do you want to go up and see if there's another question?
Yes, I can have a look.
See. It's because the chat is not. I think you have to go up.
For the, but okay,
yeah.
I don't have
Can you
to
click
stop.
on chat?
Absolutely. Oh, Laila. Hi, Laila. I know so Laila is a pathologist at our institute.
All right.
Yeah, thank you for joining, Laila.
Can you go up? I don't think we see.
Probably
everything.
not. Okay, I think there's a few other questions before. Okay.
Yeah. Have you seen?
I have a question. Okay, so this was from Hanan. I think she.
Yeah, she has a
posed
question.
Then
asked.
she was this before. One moment.
And then the question: have you been to the Serengeti yourself? Because
right.
I think earlier you said that you have a veterinarian who's been, and you're here.
Absolutely.
But you've been, right?
Yes. So I've. My predecessor from my studies, Susanna Ferreira, she kind of the project I'm working on now, she was my predecessor, so she kind of started this project within this research training group that I mentioned. She was the first a doctoral student that the ISW had for this project, and then I jumped in eventually. And her so at her time, so she did lots of field work. Then she spent, I think, in total some 6 or 7 months in total throughout her PhD. She was there. What happened when I stepped in was that actually all of the samples that we were planning on working on, they were already here in Berlin, so there was not much sample collection needed for my particular project. So I didn't go as much as she did. Actually, I went for a kind of a training course that the research group organizes every 3 years, I believe. It's coming up, I think, next year again. And I had the chance to go, but just for a couple of weeks, with a few other colleagues of mine, and also with my supervisors and a few other supervisors from my friends, from my colleagues, from the research group. So I was only there for a couple of weeks, which was very important, because in these 2 weeks I really got to know how the system works,
Mhm.
because sitting in the computer or just listening from other people is
course,
not
yeah.
the same. So yeah,
So you've
I
been-
did go for a very short time in early 2019. Yeah.
Right.
Um,
All right. I think
And
those were the questions.
I think Laila, she said,
I think maybe she answered a question someone had. I don't know. Or if
okay. Let's just scroll.
maybe it was for the if
postmortem
mosquitoes get infected by other mosquitoes.
Actually, yes. Thank
yeah.
you, Laila. That's a very good. Yeah, absolutely. So this is a very good example. The thing is, because mosquitoes, they are ectoparasites, absolutely, and they're also, we call them, most people tend to call them, because they call them vectors, because they're vectors for pathogens. And in this case, obviously, since the Plasmodium and also the mosquito, since it's an ectoparasite, they're both parasites. So there's a very good example.
Yeah,
Thank you,
good,
Laila.
a good answer.
Even though I spoke about it for 15 minutes, I couldn't remember, but that's absolutely true. Yeah. So yeah, and then she also writes, although the mosquito is just a vector. That's absolutely true.
Okay.
Yeah.
If you have any more questions, let us know.
Yeah.
We'll check the chat.
Okay.
Okay.
Perfect. Okay, great. I think I'll click on it again. So that was a bit on the about the ecosystem, and yeah, just a couple of.
Oh, that's your car.
Yeah, that's it's my supervisor and driving, and then it's two friends of mine in the car, and yeah, we took this picture when we were down there as a group. So that's our car, and so this is a what we actually see from inside the car.
That's so close.
So it is very close. So what happens is our veterinarian who's in the field full time, she just went back recently, actually, because of pandemic. She was here for quite a long time, but now she got to go back finally, and we're really happy about that, because for most of the pandemic year, we actually have virtually no samples because no one was allowed there at the time. But now she's back, and we're very happy. And Sonia is her name. She closely monitors 3 clans of 3 hyena clans, and they are used to seeing this car. They know the car. Yeah.
Okay, so they're not like intimidated or
They
scared.
they won't hide on the dens. They won't run away, somewhere else. So actually, it is very close. We got to see them from very close, and they are very easily identifiable by their spotting pattern. And also sometimes nose marks or ear marks also help identifying them, but every hyena looks different. And since Sonia, she monitors the clans quite closely and quite often, she knows every individual of the clan, right? So what happens is, and so we get, it's possible to get this close because they know the car.
Okay.
They wouldn't react, and they wouldn't be so calm about it if it was a new car. What happens is we do this, and we watch them poop, and then we drive. So usually I didn't mention this before, but hyenas, they tend to defecate in what we call latrines. So they have a particular area, not far from the den, but they have a particular area where more often they would go there and poop. So what happens is, once they do it, and we are seeing it, we quickly go as close as we can. We try to memorize where the poop is, and we try to drive a bit closer with the car and like stop it as close as we can from the poop, and very in a very quiet way, and without disturbing them, of course. Open the door, collect the poop, and come back in and drive the car a bit back again. And that's how, because all my project, I didn't say it yet, but all my project is based on fecal samples, because when we work with wildlife, what we aim at is to not disturb them, actually. So we don't want to draw blood from them or try to anesthetize them to get samples, you know. So it's possible if we do it non-invasively, which is the case of using fecal samples, and it's a recent field of studies, but it's making good progress, and many people are trying to do this and try to make as much value of the fecal samples as possible, because it's possible to do many stuff with it. So looking at parasites is one of them. Looking at immunology is one of them. It's what we're going to talk a bit about, but also it's possible to extract DNA from the fecal samples and lots of interesting things. It's possible to measure their cortisol levels, their stress levels. So that's with this very precious fecal sample, we can do many things, and that's what happens in the field. So that's the view from the car, and the cubs. So they actually
so cute.
they are very cute. So the cubs are they're very inquisitive, and they come and they chew a bit on the car wheels actually, and they play with it a bit because they're very curious about it, and they're very inquisitive. Yeah, so these are all hyena cubs. These were also pictures that I took in the field in those 2 weeks there. Yeah, so and now so talking a little bit more of what the research group does is we try to look at how so it's very well described in our hyenas, but also in many other species, that individuals are infected with different number of species, but also some individuals have very high loads of parasite. They're very heavily infected. Usually a fewer number of individuals is very heavily infected, and most individuals have quite few loads of parasites. But in fact, parasitic infections in wildlife, they're permanently there. So it's actually not so common to find individuals that are not infected. And what the study group has been working on in the most recent years is why this happened. Why do we see some individuals very heavily infected? Why some have little loads of parasites, and how in our particular case of study system, how the little crown there is their social ranking, how the one in the middle is their lifespan, how this heterogeneity of parasitic infections changes across their lifespan, and also how some energetically costly life stages, such as, for example, lactation, what impact does this have in the individuals, and actually what impact in terms of their women fitness, what impact do these parasitic infections have on the individuals and ultimately on the population, right? And why we observe this heterogeneity. So these are just a few examples of papers that dwell and touch on these three different aspects of hyena biology. And so so we it's this kind of a triad that we are interested in the life history of these hyenas, and as I mentioned before, it's very easy, and since we're collecting fecal samples quite often, and since we know all the individuals, this is the perfect study system to collect many different samples across their lives, right? Because we can sample them quite often. It's a big problem in wildlife research that you get this chance of sampling a particular individual, but it's actually very hard, if you think about it, for most species to resample the same individual, right? And so this is why our study system is actually ideal, because we can, in a non-invasive way, which is very good because we want to disturb them the least possible. So we can track them throughout their life and check not only collect the samples actually, but also check how they are doing in terms of survival, in terms of reproductive success, how many cubs they have, each female has, and so all these life history data that we can collect from them is very beneficial when we then try to interpret our results. And and then we're also interested in my particular case, that's kind of the purpose of the project, in their immune response, because obviously mounting an immune response against these parasites is costly, and it perhaps it takes resources from other activities that they would also do. So it's kind of exploring, the project is all about exploring these triad, and the immune response part is kind of where I stepped in when I joined the group, and so our question was, and so I think you've realized by now that we're mostly interested in gastrointestinal parasites because we collect fecal samples. So we came up with this question whether actually it was possible to measure local gastrointestinal immune response. And we came up with these three main immune effectors or immune markers that tell us a bit different parts of the immune response. So they're colored, and mucin is already very important, for example, in the gut, in the very early stages of parasite attachment, when the larvae attach to the gut. And already then mucin production increases. We can see that on the light purple rectangle, and then secretory IgA. IgA is an antibody, and IgA is the most common antibody in the gastrointestinal system. And for example, IgG is more common in the systemic, in the bloodstream, but we're interested in the gut, and so we also wanted to look at IgA, because in the gut IgA is the most important one. And we can see on the right side of the picture, on the orange square, that antibodies are also very important in also the later stages of worm expulsion and infection resolution. And also more recently, we came up with trying to also measure neuropterin. This little red rectangle is marking macrophage, because neuropterin is a product of macrophage activity, and macrophages are phagocytic cells that are very important not only in the response to the infection, but also actually in the tissue repair after an infection happened, because obviously the epithelial cell turnover is very quickly in the gut, happening in the gut, but also when there's tissue damage, macrophages are very important for helping this tissue repair.
And are
after
there white
an
blood
infection.
cells, macrophage?
Yes. So yeah, exactly. So
people might know them, right?
Yeah,
like white blood cells.
I think so. Yeah. So they are they're what we broadly call, so they're the experts on phagocytosis. That's where the name comes from. Exactly. Yeah. So this is kind of a we if there's questions a bit more into detail, but I don't want to go into detail on
we also have 20 minutes left.
Okay, yeah. No, that's perfect. I think we're very good on time. So, and this is a bit of what happens. So I already talked a bit about the feces collection, and then so they are shipped to Berlin, and then we keep them at -80 degrees in very cold temperatures. And then what we do is we freeze dry the sample. So we kind of remove all the water, and then we prepare these extract solutions that we can then do our immunoassays with. And that's the extract. The final extract is the little picture on the right there. And yeah, so these are kind of the assays that we do. Most of them are quite so actually all of them are quite straightforward immunology assays. The question is, they needed to be validated for our study species, because not many people, well, actually virtually no one had tried performing these assays with fecal extracts from hyenas. So you always have to, when you set up a new assay, you always have to properly validate it for your study species. So I also won't go into too much detail on the assays, but then obviously if there's questions, we can talk about it. They're mostly ELISA assays, or in the case of lysozyme, we have this diffusion agar diffusion assay, where we put bacteria in the agar and then we check how big is the halo and what's the lysozyme concentration of the feces. So that's just to give an idea. And also we can do interesting stuff.
What is that?
Yeah, it's so this is a histology picture. It's stained with this EPCAM staining that stains the surface of epithelial cells in brown. And what we can see here is actually from the mucus layer of the feces. So since the feces are collected very fresh, it's sometimes possible to actually remove and collect or store in formalin the mucus layer of the feces, and then we can do histology on it. We can do it's very pretty. Okay, yes, and we can do histology on it. We can also do cytokine expression assays on this, and yeah, so it's just something else that is possible. And yeah, so I also, concerning the results, so this was a paper that we published recently this year, and it's also open access, so you can have a look at it if you're more interested, and all the assays are neatly described in there. And so this is just an example of some eggs that we look at, and actually there's a little larvae on the lower right picture also that we see, and the graph shows that actually on average our hyenas are infected with 2.5, something between 2.5, 3 different parasites, or different genus of parasites. So you can see this. So yeah, it's actually, the norm is that they have more than one different parasite.
Interesting.
Yeah,
okay.
so having less or having only one or even 0 parasites, you can see that it's not so common as having 2 or 3. Yeah, so that's just, yeah, and I will probably go very, very quickly on these last slides, because it's so we can, interesting stuff we can do is to look at how the different parasite community from adults and juvenile looks, for example. And so that's different, the names are just different species names, a parasite species. And so you can see that, you know, the juveniles, they have, they're quite different, and you can see this interestingly, and then we can see also they differ on their juveniles and adults on their levels of mucin, for example. You can see on the left graph, and on the right graph, you can see that also, so on the x-axis you have Ankylostoma egglode. Ankylostoma, it's a hookworm, it's the most common parasite that we find. And you can see also that as the load of the parasitic load increases on the x-axis, the mucin levels also are different if they're juveniles or adults. And so this is all interesting stuff that we can do, and we can create models and plot this interestingly. Also, mucin, for example, increases depending on the parasite richness, which is the number of genus that they're infected with. So yeah, and we can do also very interestingly on the right, on the lower right graph, we can check actually we can do longevity and survival studies because we know how long they live, and we can, for example, see that the individuals with in this case with low IgA secretion, they tend to survive more than individuals with high IgA secretion, because the blue, which is the high IgA concentration, you can see that the survival line, this is a survival rate on the y-axis, and you can see it's going down way quicker. So we can do this. So this leads us to suspect that this is possibly costly for the individual to actually produce produce IgA, because the ones that are producing more, they tend to survive less. So and these were just a few examples, but the paper is more, it's very detailed, and people, and it's open access. So
if people are really interested, they can look it up.
So just to finalize, going back to the question at the beginning, how many parasites are too many parasites? It's actually, the answer is a word that it's an answer that we as scientists don't appreciate so much, but so it's actually that it depends.
Oh no,
which is
always that answer.
It's always the same. And it actually depends, because species that are more harmful than a lower number of parasites, actually that's already too many parasites. A fewer number of individuals cause bigger problems, whereas if it's a species that is not so harmful, then you can have many parasites, and it's not so much a problem. So it really depends on the species. I know it's
always the answer.
I know it hurts to finalize with this. And just as a conclusion, I really like this sentence from George Shaw, which says, "Science is always wrong, and it never solves a problem without creating 10 more." So
yes, we heard this right now.
Exactly. So that kind of concludes a bit of
so
what I had to say.
I learned a ton about parasites.
That's
thank
good.
you. I thought that was
uh,
really interesting. So thanks so much for that.
Thank you.
Let's see if we have some questions. Maybe go into the chat, because I also have my own questions, of course.
Okay.
There we go. There's chat.
Let's have a look.
Okay. Can you maybe you have to scroll up
I
a little
think
bit
we have to scroll up. Okay.
Oh we got a nice reply from someone asking some questions. Thank you. But we have a question underneath. It says, when you collect samples, do you have to worry about getting infected by parasites?
So technically, no, because we are very quick, and we also obviously we are using we are using gloves,
protective
and
gear,
protective gear, and we put the sample on a nice a plastic bag and all this, and then we preserve it. So not directly from the feces itself. And actually all the parasites that the hyenas have, none of them is really zoonotic. None of the species that they have. So not directly, but then also, there's always the issue, and you mentioned it on your stay in US, that it's field work. So there's ticks around, and also closer to the dens, it's not so unusual to see also fleas, for example. So since the cubs are staying there most of their time, it's a good environment for the fleas to replicate and to live. So actually, there's fleas around, and there's ticks. So you never know. So you never know what you're going to get from them, but not directly from the fecal sample itself.
All right.
Exactly.
I have a question about how you first felt about working with parasites. Do you find it scary, or were you just kind of like, oh, whatever, it's just fine?
Right. Absolutely. No, so definitely not scary at all, because I always, already since the veterinary studies, I was very fascinated by it. So and we always, it's for me, it was always the thing that, I have this funny story to tell at home. When I learned about this interesting stuff from my teacher, I always thought, not so much scary, but very fascinating, very interesting. And so
so it wasn't like a big shock.
It wasn't not at all. No. It wasn't a big shock, not at all.
All right.
No, I was prepared for that.
All right. I wanted to stop sharing our screen.
Oh yeah, we
actually,
can.
so people can see us better.
True.
But
We
I
can
think I
do-
just have to click on
Yeah.
stop. All right, people can see us. Here
Cool.
you go.
Yeah.
Okay, so it was
Cool.
just like, oh, I think this is cool already. So you don't.
Absolutely. No.
So not like how I would be.
Yeah.
I would be like, oh,
No.
okay, give me the hyenas, but
Of
keep the
course.
parasites
Yeah.
away.
No. This was obviously one of the reasons why I thought when I saw the application for the project, I saw it was parasites, and
Sign me
I
up.
went for it. Yeah, exactly.
And did you ever have any scary encounters with wildlife? Did you ever think like, oh, there's a lion over there kind of checking us out?
Yeah. So during these 2 weeks we were there, absolutely there were lions. There was a lioness who actually had a cub, and we could see it from the house we were staying on, but we so not directly. That was not scary because they kind of kept their distance, but we could still see them. But we were careful, obviously, when walking around.
Mhm.
But we did see from our so we had it was a morning where we were having breakfast, and we actually saw a group of lionesses trying to hunt down a buffalo.
Okay.
And so, but that was quite far off.
Over breakfast.
So
though?
over breakfast, it was
Seeing
very
that.
interesting. Yeah, and we were far away, but that was very exciting. They failed it, so the buffalo
failed,
eventually
yeah.
got away. They failed, but it was probably the most exciting experience. But scary encounters, definitely not. No.
Okay.
Yeah, it's also very, when you're driving and you pass by a buffalo, it's very,
Pretty big,
when you
no?
do it's for the first time, it's like a big, it's very impactful.
Yeah.
It's lots of respect. They're very big, but definitely not scary.
Nothing too
No,
scary.
No.
Okay. So you
Yeah.
said that you touched on it a little bit how you couldn't or some things that have changed, but
Yeah.
the current situation with restrictions, has it changed how you work in the lab? Like I know you couldn't
Okay.
like go to Tanzania or visit the national park, but
Right.
have there been restrictions that have really limited in how you do lab work, or has it been okay?
Yeah. So what happened was, as I suspect in many other labs, we kind of had to take shifts or make sure that we would schedule our experiments, and at days where we could just schedule with other people. Luckily, in our case, our immunology lab, it was quite easy to coordinate all this because at the time there were mostly 3 people working there, and it was not that complicated.
Okay.
But we tried, especially during the hard lockdown, we could still go to the lab. The labs didn't shut down, but we really had to schedule very strictly who was using it at what point. But I guess it's just a matter of coordination.
Right. So like
Yeah,
time management.
absolutely.
It's just-
And I guess this was the case for most
Okay.
labs.
I always have 2 questions
Yeah.
I love to ask because
Okay.
I
Go for it.
find the answers are sometimes very similar and then sometimes they're very, very different. So I'm going to ask
Okay.
both of them, but
Yeah.
I'm going to start with what is the worst thing about your job?
What
is what you could live without? What is annoying? What what is not your favorite?
So the worst thing is probably, I think at this time a bit more, since now I'm mostly I'm writing or finalizing data analysis, and this means you're sitting at your computer, the whole day. So that's definitely the worst, is that when you do research, even if you have lab work or field work, there's always a point when you're going to come back and sit at your desk, right?
Yeah.
There's always that point, and I dread that point, and that point is now
The
actually,
writing part.
Yes.
You want to do the experimenting part. I see.
Exactly. So yeah, it's not so much the writing itself that is painful, but or the analysis that's fun, but it still forces you to sit in front of the PC.
Okay.
And that's kind of I would say. yeah.
Okay. So what is the
Like,
best part though?
Yeah.
What is the best
that-
part about your job?
Right, absolutely. So it's during my PhD, what I love the most is really that it's every day can be so diverse, because there's sometimes when you're in the lab, but even if there's some days that you can start in the lab and then you are ending up your day already analyzing data.
Mhm.
So and you can, I liked that you can schedule your own day as it kind of pleases you the most,
Mhm.
in the sense that your day can be, you're doing the lab for, I don't know, 3-4 hours, but then there's an interesting paper that came out, and then you're reading this paper and looking careful into this, and then you end up your day,
Mhm.
in our analyzing your data.
So every day is a bit different.
It's a bit different, yeah.
Mhm.
So and I like this that you're changing activity, and then suddenly an important email comes in and you need to shift your focus a bit.
Mhm.
For and I like this this constant change of
Yeah.
your daily routine can be very different.
Everything that's not
Just-
writing.
Yes.
You're
Yes.
like, oh, I have to
Exactly.
do writing, but there's
Yes,
this email.
but there's
I have
this
to do.
email, right? Exactly.
I love that. I love
Right.
that answer.
Exactly.
And
Exactly.
what do you see yourself doing next? I mean, eventually,
Oh.
the PhD comes to
Wow.
an end. It
Yeah.
has to. Do you want to stay in research, or
Right.
what comes next?
Yeah. So I think if you would ask me this question 3 weeks back, my answer would be, I have no idea what I'm going to be doing next. Although I really want to, keep doing research.
Mhm.
But actually, quite recently, I got a job offer.
Oh.
Yes. So I'm going to be shifting my. So I'm not going to be moving on to postdoc or something. So I'm not going to strictly stay in academia or doing research, but I'm going to be, since next year, beginning of next year, I'm going to be doing only actually pathology work.
Okay.
Because I got, so it's this position in Edinburgh in Scotland.
I love that place.
I know, I love it as
Yeah.
well.
I see you moving away.
So I'm moving, but still. So and it's a clinical position.
Mhm.
So I'm going to be dealing with companion animals, farm animals, but also wildlife, which
Okay.
I'm
Your
very
biggest
very
love.
excited about. So I think that's also partly why I was selected for this position. But I definitely want to keep my feet on research.
Mhm.
So it's not going to be full time as it is now doing research, but I definitely want to publish the more interesting cases and this kind of stuff. I definitely want to keep in touch with research, because
Right.
I like this this outreach that you can get by making a study available
Yeah.
for people. And so I want to keep involved in research, but I'm not going to be doing it full time anymore in the and so who knows what comes next. Perhaps I go back to academia, and but at the moment this is but it's just very recent. Yeah, if you'd asked me this
Congrats.
3 weeks ago,
And I like that you
this-
share this with us, because it shows that
Yeah, thank you.
what you can do afterwards. Like, you don't need to do
Absolutely.
strictly one thing for the rest of your life. You can
Absolutely.
like diversify
Yeah.
your experience. And
Absolutely.
Edinburgh is
It's-
one of my favorite places to go. So
yeah.
I'm very excited
Well,
for you.
Absolutely.
And
Yeah,
we're planning
thank
to. I want to take my family to Edinburgh because I love
Right.
it so much. And
Okay.
my former roommate just moved there
Oh,
and
wow.
doing a
Okay.
PhD there.
Oh, okay.
And I want to visit, so maybe I'll
Okay.
hit you up and
Yeah.
be like, hey, let's take
Of course.
let's talk
Absolutely.
about parasites and stuff over a cider.
Yeah.
But that's really
Absolutely.
cool.
Yeah, I'm
Congrats.
very much looking forward. And
Yeah.
I think, to be honest, I think I studied something that is so diverse that you can do. Yeah, you can there's also, you know, food security.
Big
There's people that doing food security, there's people doing only clinic, there's people you know, doing only lab. But yeah, so with the veterinary background, you have all this range of possibilities, and it's also kind of easy to, you know, jump in from one to the other. And I like this and I'm very happy I did studies on such that then allow me to just do so many different things.
And
right.
thanks again for being and answering all these questions.
Yeah,
Thanks again
no,
for asking
thank you
the
for your
questions.
questions also.
Super exciting.
Yeah.
And I'm excited to see what you do next.
I'll keep in touch.
You promised you would do a Twitter, so
yes, I will.
Eventually, people will be able to
now
find
it's
you on
public.
Twitter. Now people
I
know.
have to do it. Absolutely.
And thanks again for joining us. And if you have any more feedback for us, we have a little evaluation that we're going to post in the chat again, that we can tell us how you liked it and what we can improve.
Oh, I'm being evaluated here.
Yeah, I mean, mostly me, I think.
I didn't know that. Okay.
I'm already excited that you're here, so you're already winning.
Good.
Okay. And next month, we have another Kaffee Klatsch. That one is in German. And it's about rivers. So we never had someone talking about rivers, and it's about the last wild river in Europe. One where not a lot of like populations or people are actually even living around that.
And depending on the river.
And it's a very specific river that she studies. Her name is also Franziska. So it's going to be a very funny, like, oh, fancy. So we're going to talk about that. And she does a lot of citizen science. So people getting involved also in science, which I think is very cool. And that is November 7th, I believe, the Sunday. And it happens during Berlin Science Week. So there's a lot of stuff happening already.
It's coming soon.
It's going to be a very busy week for me, but I'm finishing strong with another Kaffee Klatsch. And I'd hope for you to join us again. And thanks again for being here.
Thank you,
such a great time. And we'll keep in touch. And I hope
thank you so much.
We'll see you again. And have a great Sunday. I know the weather was very nice. And hopefully next time it's still going to be nice enough. We'll see. But we'll be here anyway. And see you then. Thank you very much.
Thank you so much, guys. Thank you for joining. And thanks again for having me, Franziska.
And have a happy Halloween.
Very important.
Very important. Bye.
Ciao. Ciao, everyone.